Let's get much more intimate with our focus, if we may. Katherine Luzuriaga is our next guest, physician, researcher. She's a professor of molecular medicine and pediatrics at the UMass Medical School. Her research focuses on, talk about disadvantage, on developing ways to prevent or manage HIV infection in children, including very young children, infants born with HIV. In 2012, with fellow researchers Hannah Gay and Deborah Persaud, Luzuriaga developed a functional cure for an infant infected with HIV by starting an aggressive treatment within 30 hours of birth. These doctors were able to effectively eliminate the virus from the child's body. That infant is now 2.5 years old and showing no signs of HIV. That is pretty fantastic. Luzuriaga, along with Gay and Persaud, were named to Time Magazine's 2013 list of the 100 most influential people in the world, right up there with Gabby Giffords and Pope Francis and Jay-Z, no less. Please join me in welcoming the wonderful Katherine Luzuriaga. Katherine.
Thank you. I'd like to thank Chancellor Motley for inviting me to be here today, and I'm just very honored to be in this group of wonderful people. So what I'd like to do today is to share with you some of the successes that we've had around treating pediatric HIV infection globally, but also some of the remaining challenges. And I'll have to say that as a physician and scientist, it really has been my privilege to participate in this. So, just a couple of statistics on pediatric HIV infection. In 2012, there were about 3 million children living around the world with HIV infection. There were 260,000 new HIV infections in children, and about 200,000 children died. Now, mother-to-child transmission is the primary mode of infection, and infection can occur during gestation, or as we say in utero, during delivery or postpartum through breastfeeding. I told you that there are about 260,000 babies that are born each year with HIV infection. This is a global map of where those infections occur, and as you can see, in North America and Europe, there are very few children born nowadays with HIV infection. And that's because over the 30 years of the epidemic, we have come to learn about, we've worked very closely with our basic scientists to learn about HIV pathogenesis. Antiretrovirals have been developed based on that knowledge, and we've been able to bring them into the clinic. So if moms now are identified as being HIV infected, then we can give them antiretrovirals to preserve their health and to prevent HIV infection of their infants. So HIV infection in infants is a very rare event, luckily, in North America, Europe, and other high-resource settings. But as you can see, the vast majority of HIV infections now occur in low-resource settings, such as sub-Saharan Africa. And this provides some real challenges. We have the science to prevent most pediatric HIV infections now. The issue is really crafting the approaches for lower resource settings that are effective in lower resource settings. So this is a very complex view of the HIV life cycle. The point here that I want to make is that there are various steps in the virus life cycle. So the virus enters, and it has to, it's a retrovirus, so it has its nucleic acids are RNA, single-stranded, has to make double-stranded DNA. That gets integrated into the host genome. We have lots of antivirals that block the viral life cycle at many different steps. However, we aren't able usually to get to individuals very early in infection, and so the virus has an opportunity to make these double-stranded DNA and integrate that DNA into the host genome, and that's the barrier to cure. So if you treat a patient, you can reduce the amount of new virus that's produced, but you don't flush this virus from the host cells. So that's why, to date, cure has been so tough. And in fact, in over 75 million people that have been cumulatively infected with HIV since the beginning of the epidemic, there's been only one real cure. And that was this gentleman, Timothy Brown, who is also known as the Berlin patient, who was HIV positive and developed leukemia, for which a bone marrow transplant was indicated. And what happened was anybody who has leukemia undergoes a transplant, undergoes irradiation of their bone marrow and immune system, and also receives very high doses of chemotherapy to kill all of the cells of their immune system. And then he received a bone marrow transplant. Now, interestingly enough, the transplant physician realized that there are certain individuals who harbor a genetic defect such that they don't express a protein on the surface of their cells that's necessary for HIV entry. So when this man needed a bone marrow transplant, this transplant physician said, well, what if we transplanted this person with cells from an individual who is resistant to HIV infection? And that's what they did. And the net result, this is a very, very complex slide, but the net result was many years later, this patient remains without HIV rebound. Now, if you use very, very sensitive tests, you can detect very tiny traces of HIV in this patient's blood or immune cells. And the real question is, you know, since we're really pushing the envelope in terms of the sensitivities of these assays, are these false positives or are these actual viral footprints? The bottom line, though, is even if these are viral footprints, they don't appear capable of reigniting the rounds of viral replication that are necessary to allow HIV to return. And Timothy has remained off treatment and remains pretty well. So what have we learned from this patient? We learned that replication-competent HIV reservoirs can be cleared, that is, those integrated viruses, and long-term virologic control can be achieved, but persistent low-level detection of this HIV DNA in the host cells doesn't necessarily predict relapse. So how can we apply this to children? Well, in Africa, you know, bone marrow transplants are really expensive. They cost hundreds of thousands of dollars. They also have high morbidity and mortality. Turns out in Africa, HIV also has very high morbidity and mortality. About a third of HIV-infected infants will die by their first birthday, half will die by their second birthday, and 75% will die by their third birthday. So that means if we really want to make a difference in the lives of HIV-infected children, we really have to come in early. And that's what we started to do way back in the 1990s as we began to get more and more antiretroviral agents. We began to combine 3 and 4 agents together and started to try to give them as early as possible. And these are results of a study that was started in 1995. This is age on the x-axis, RNA on the y-axis. And what it basically shows is that once young babies under 3 months of age are treated with antiretroviral therapy, you can control the virus, and these children have lived anywhere from 14 to 18 years now with very strict control of viral replication. But as I told you, the antiretrovirals can control the virus. These kids are growing up. They're, the 2 oldest just graduated from high school and have gone on to college. They are leading ostensibly normal lives, but they still harbor HIV. This approach has been used in young children, and a randomized clinical trial in South Africa demonstrated that early treatment, so this is probability of death and this is age, and basically there was a much lower probability of death with early treatment, which was treatment before 6 months of life, than those who had deferred treatment. So early treatment now is the recommended standard of care, but as you can imagine, in the absence of a fully functional healthcare system, that's often difficult to deliver, but it's something that we're really trying to work very hard on. Now, HIV treatment not only makes a difference in terms of morbidity and mortality, but what we are starting to learn is that in those very, very early days to weeks following HIV infection, maybe a very, very crucial period of time in which the virus gets into host cells and in particular populates some very long-lived, what we call memory cells. So these are cells that we generate through vaccinations or exposure to infections, and these are the cells that provide us with immunity to that particular pathogen for life. Turns out that babies are not particularly good at making those cells. So we ask the question of whether that inability to make those memory cells might make it less likely for HIV to establish these long-lived reservoirs. And in these early studies that we've done, what we've shown is that babies who were treated before 6 weeks of age, those are the red circles, had much lower levels of these memory T cells that were populated with the HIV genome than children who were treated after 6 weeks of age. So this begins to tell us that there may be this window of opportunity that we may have if we are able to come in early to be able to clear the infection. And we're starting to get similar signals from adult studies. There are a number of studies here that say that if you come in really early, you may again be able to restrict formation of that very long-lived memory T-cell reservoir. So this is really pretty encouraging, and there are a number of different trials now that are being proposed around the world to test this. But as we were gearing up to start to try to do these trials, serendipity happened. And about a year ago at this time, I received a call from a very long-time colleague, Dr. Hannah Gay, who practices in Mississippi. And she often calls me, or we've talked to each other over the years when we have particularly difficult cases. She told me about a baby who at that point in time was 2 years old, was born a little bit early to a mother who was not known to have HIV infection. That is, she hadn't engaged in prenatal care, wasn't screened, didn't receive antiviral therapy. The delivery was really pretty rapid, so they couldn't get antiretrovirals into mom or baby. And the baby was born in rural Mississippi, transferred to the University of Mississippi Medical Center by 30 hours of age. Hannah basically drew what we always draw, which is some bloods to see whether the baby was infected, and immediately started antiviral therapy. And what she started was a 3-drug high-dose regimen because she was very concerned that not only did this child have a high risk of infection, but if this child was already infected and she treated with 1 or 2 drugs, that she might generate resistance to the virus. And basically the mom's infection was confirmed. Unfortunately, the baby's blood tests came back in the first week of life, indicating that HIV nucleic acids were detected in the baby's blood. So that means that the baby was infected. The baby did meet criteria for standard criteria for HIV infection, and so antiretroviral therapy was continued. HIV was detected 3 more times in the peripheral blood, so there was no doubt that this baby was infected. But then with the antiretroviral therapy, the viral load in the blood fell below the detectable limits of the clinical assays and has remained there through 36 months of age, so 3 years of age. Now, what's interesting is that, unfortunately, I think, as was alluded to by the prior speaker, we have areas where this particular mother had a lot of social difficulties and wasn't able to make it into the clinic for a period of time. So she stopped coming to the clinic at 15 months of age, and Dr. Gay made a lot of efforts to get her in, finally got her back in at 23 months of age. And at that point in time, the mother reported that she had stopped treating the baby at about 18 months of age. So by the time Dr. Gay saw her at 23 months of age, the baby had been off treatment for 5 months. And what Dr. Gay fully expected to see was that the virus would come screaming back. Did the routine assays, got ready to put the baby back on meds when the viral load came back undetectable, which was really a surprise. We never see that. So she said, well, maybe that's a lab error, and repeated it. And again, everything came back negative. So she gave me a call because she said, I must be going crazy. There's got to be something wrong here. And I said, no, Hannah, I think you might have treated the baby early enough to be able to achieve remission. And that's indeed what we think she did, because she's been tested repeatedly now through 36 months of age. On routine assays, we have not detected anything. Again, we've put this baby through an exhaustive panel of ultra-sensitive assays that are research lab based, and we have a couple of signals in the white blood cells saying that we may again be able to detect viral footprints, but again we are not detecting, the virus has not come back, and the baby continues to control the virus. So again, and the baby has normal CD4 T cell counts, responds well to childhood vaccines, otherwise seems to have a very normal immune system and is growing up and doing quite well. So what we think we've done with this baby, this was serendipity, but we've, through intensive study, what we think we've done is to, by starting immediate antiretroviral therapy, is to markedly limit the size of the pool. But as I told you, again, babies have very low levels and a reduced ability to make these memory cells that HIV likes to hang out in. And so what we've also not only reduced the size of the pool, but we think that we have limited the number of these long-lived cells that are infected, and we think that's why this very early intensive therapy was successful in clearing the virus. Luckily, we have a very strong medical research infrastructure here. The NIH supports an HIV Clinical Trials network focused on prevention and management of HIV infection in women and children, and so that has enabled the rapid development of a study that will attempt to replicate this case in other children. This study will be carried out both in the US as well as internationally, and we very much hope to be getting started on this by the beginning of the year. So I think I'll stop there and I think many people know that anything in science takes a team of, often takes a large team. So Hannah Gay, Deb Persaud, the children parent, the mom and the child that allowed themselves to be studied, as well as all our laboratory. And then finally, this would not be possible without funding from a lot of different sources, including the NIH. Thank you.
Thank you very much. Thank you, Katherine. Katherine Luzuriaga, thank you very much. Wonderful, wonderful. That is amazing and deeply important work, and we're very grateful to you for being with us today. Just fantastic. Tweets are flowing in for you. Oh. Peggy says, thanks to Rockstar Doc Luzuriaga for presenting on the devastation of pediatric HIV. I've got a lot of questions, but let's go right to Loves Life 28. Who asks, what inspired you to take on this issue, HIV in children?
So, I've always been interested in microbiology, infectious disease, and that's why I decided to specialize in that. But I think what inspires almost any physician scientist is going to the clinic. And in those very early days, we just had too many babies that were dying. And it's really tough.
What clinics, where were they that you were going to, Katherine?
So, late 1980s, I did my training out in Worcester, at UMass Worcester, and so unfortunately, right we began to see, we began to see a number of women who were infected, a number of children, and because the children have such a rapid tempo of disease, they were often the first in the family to present. So again, I think just going to the clinic, seeing these families, not being able to do anything to the children, and having fantastic training allowed me then to begin to ask some questions along with my colleague John Sullivan, and to go back and forth between the clinic and the lab to try to answer them.
That means the answer really is just human compassion. When you saw them, you wanted to do something.
Well, I think absolutely. I think that it's hard to practice medicine without a measure of compassion.
Amen. Amen. I have a layman's question, and maybe I should know the answer from what you just described, but I can't say that I do. Is this essentially the same treatment as the antiretroviral cocktails for adults, that Magic Johnson and lots of others are taking, or is it something else for the infant?
Yeah, so it's the same treatment, but again, when you start treatment in a patient that has been infected for a while, you're controlling the ability of the virus to make copies of itself, but you are not flushing the virus from the host genome. So, what we're trying to do now is to look for ways to flush that virus.
So an antiretroviral can actually get in and change the genetic sequence in these memory T cells, or am I totally go back to school, Tom?
No. So
I'm really out of my depth.
The antivirals are blocking the ability of the virus to make copies. So they're blocking that viral life cycle.
I see. So that's
but
the intervention.
They don't change the genomic.
What about that? We had on the show yesterday. We had Craig Venter, the famous, a biotechnologist who first sequenced, along with others, the human genome, and his big thing now is synthetic life. Take the cell, strip out its original genetics, drop in new engineered genetic code, and you've got a new thing going on. Is that an angle of approach to this problem, or people?
Wow, I like that. It's not something
it's like
that's
taking
been
the engine
proposed,
out and dropping a new one in
that's a really interesting proposition.
Okay, maybe we go there. Questions for you. We have such a high incidence in Africa.
Yeah.
And I wonder how the African leaders that you're dealing with there look at your campaign, your crusade, your work. I mean, we had a population specialist on projecting out, by the end of this century, Nigeria will have a larger population than China. That's the projection. And here you are asking for pretty high intensity bone marrow treatment and so on of infants. Are they, rallying to this cause, or are they like,
so I'd like to say that we are not proponents of bone marrow transplants or anything, and so what we have really tried to do is to craft treatments that are appropriate for lower resource settings, and we think we can get there.
But you take my point, in their
so they've
pyramid
been
of
very
concerns,
supportive,
they
because anything that affects, in some places 50% of women aged 15 to 49, which is your prime workforce, are infected with HIV. And then if you've got significant numbers of babies that are being infected with HIV, that's a real issue for the economic development of a country. So a number of countries that are most hard hit are very interested in working with us to craft responses to the epidemic.
Katherine Luzuriaga, intellectually, scientifically, emotionally, your work really moves me. I'm sure it moves all of us. Thank you very much for being with us. Thank you, Katherine. Thank you so much. Thank you. Katherine Luzuriaga, and HIV treatment for the youngest of us. Thank you so much.